Thank you very much. It's really an honor to have been selected for making a presentation here. I'm extremely happy to have become a part of the Lincei family. And I'm going to try to tell you a little bit of what we do connecting nanoscience with the biomedical field. So maybe I can start with a disclaimer that what you see here in the background are not galaxies. It's actually, we are moving all the other way in the scales. So from the 10 to the 9th, we go to the 10 to the minus 9. And we are talking about nanomaterials. And the reason that we use nanomaterials is that the properties of essentially any material change drastically when you go to the nanometer scale. And not only they change, they can actually be modulated when you are able to modulate the size and the morphology of the material. And so here we focus mainly on plasmonic nanomaterials. And again, here we will have plasmas, but these will be plasmas that only involve electrons, and they can be created at room temperature and pressure, standard pressure. So very simple materials. Now, why is this important? Well, you have all probably seen many predictions of the applications of nanotechnology. This is already another old picture. I found it in a presentation which was posted online in 2006. It's probably earlier than that. And so there were already many predictions of what nanotechnology will do for society in the future. If you look carefully, I'm not going to read everything because we don't have so much time. Most of the foreseen applications that you can see in this slide are already in our daily lives. So even though many people say that nanotechnology has been a hype, I disagree. Maybe many things have been oversold, but most of the applications that you can see here are on our cell phones, essentially. Now, let's go a little bit back in history, because this is not all new. And so, in fact, you can already find nanotechnology, even though it wasn't called like that, in the early Roman artwork. So this is the famous Lycurgus cup, which unfortunately is not in Italy. It's held in the British Museum in London. And it's an obscure piece of art that is hidden in the, if I remember correctly, 5th floor of the British Museum. And they hold it on a shelf with a lamp behind, which is switching on and off intermittently. And then depending on whether it's on or off, you see a color change in the glass of the cup. And so the scientists at the British Museum took a little piece of the glass, put it into an electron microscope. This would be the equivalent to the telescope in the nano world, which allows us to see down to the nanometer scale, so millionths of a millimeter. And they actually found many particles like this with a scale of 50 nanometers, so 50 meters to the minus 9. And these are made of gold and silver. And so these colors that you see here are golden colors and silver colors. And the same thing you can see in many stained glasses in churches and also in many buildings anyway. So not all of them are made of nanoscale metals, but many of them are. And this is why you can still see the colors over centuries after they have been made. Okay, so this has been very important already for applications in staining. Now, scientifically, we usually put the start of the field of nanoscience related to plasmonic colors in 1856. 1857 was the publication, and it dates to Michael Faraday, the same Michael Faraday you know for many different things. And so Faraday was a very curious man, and he was actually working routinely changing topics. And actually, he was very recognized already at his time. You can see here a reduced list of the academies that he was a member. Lincei is not there, but we see Florence, Bologna, Modena, and there is an et cetera, that maybe includes Lincei as well. Anyway, so one of the studies of Faraday, already closer to his death, quite premature, was actually trying to understand the undulatory theory of light. And he wanted to see whether he could demonstrate the relationship between the state of division of metals with their interaction with light. And actually, again, I recommend that you read the manuscript. It contains also many elements of how science should be done that should be still valid today. And in part of this article, he refers to diffused particles of gold, describes its production by using chemistry, the relationship between size and color and aggregation state, and some other changes. So this is actually also a sort of prediction of what would come in the field of colloid science. And some of the samples that Faraday produced in the 1850s can still be seen at the Faraday's Museum in London, and you see a picture here. And he actually talks about the gold forming part of exceedingly fine particles, which becoming diffused produce a beautiful ruby fluid. So this would be water, essentially, with a collection of very fine particles of gold that provide the red color that we could also see in the Lycurgus cup. So this is the first scientific description of why gold can be red. Now, at Faraday's times, there was no availability of electron microscopes or even spectrometers, so he didn't know that actually the crystalline structure of gold in the exceedingly fine particles was exactly the same as in bulk gold, so nothing to be assigned to changes in the crystallography. And the red color actually came from a very fine peak around 520 nanometers, about 2.3 electron volts, that is extremely efficiently absorbed and even scattered light by these exceedingly fine particles. And this is what causes the red color. So about 100 years later, the same type of material was applied for the first biomedical application of nanotechnology that I'm aware, which is the pregnancy test. The pregnancy test is based on using this type of gold colloids, which are made by simply reducing gold chloride with sodium citrate. After a few minutes of boiling, you actually see the formation of the famous red ruby color that Faraday predicted. And this is connected to particles that look like this, about 15 nanometers in diameter. And they are used to link to antibodies that selectively recognize the pregnancy hormone. And this is what actually provides the color in the test line or in the control line. So the test line only if there is pregnancy. And this was sold by millions of units since the 1960s. And this is exactly the same concept that we have all used in the past few years for the COVID test. All right, so gold nanotechnology has been around for a very long time, back to the Romans, but also in the modern times. Now, what is the scientific basis of these colors? This is connected to the plasmas. And so if you have a bulk metal, you can produce charge density waves by illuminating with a specific wavelength that is called the bulk plasma wavelength, characteristic of each metal. If you have a very thin film, the electrons are not able to form a wave inside of the metal, so they are confined at the interface. If you have a nanometer-sized particle, the waves cannot travel, so you have a dipole that is periodically oscillating in resonance with the frequency of the electromagnetic radiation. One specific for each metal, for each particle size, for each particle morphology. I'll show you in a minute. So depending on which metal you're creating in the form of nanoscale particles, you can have the red gold colloid or the yellow silver colloid. So here you see again colors that are assigned to metals, very different from what we are used to in the bulk, in the macro world. Particles look like this. Another effect is that when you illuminate these particles with a specific wavelength that excites this oscillation of electrons, you also create a very large electric field at the surface of the particle, which will be essential for the applications that I'm going to describe in a minute. Now, if you change the morphology of the particles, then you can actually excite oscillations of electrons either along or across the long axis of these, what we call nanorods. And so depending on the ratio between the length and the width, you can actually select the wavelength at which these particles are going to absorb and scatter light, and this will be seen also in the color of the dispersion of these particles. But it also allows you to go and select the wavelength that you want to apply for a specific use of the material. So for many years, we and quite a few other groups in the world have been working on making gold and silver particles in different sizes, different morphologies, with very high quality. And we define quality as how similar the particles are to each other within a single sample. So what you see here are electron micrographs from droplets of a colloid that has been made by chemical colloidal science, where the particles look like this. They are all very similar to each other. They all have properties that are similar to each other. And this is what allows us to define what is the wavelength that we are going to apply. The color images that you see here are either experimental or theoretical demonstrations of how the electric field is created and confined at specific spots depending on the morphology of the particles. And why is this important? This is important because then you can also select to operate at what we call the biological transparency window. You can see here, this is the spectrum of human tissue. And it typically absorbs and scatters light in most of the visible. But if you go to the near infrared, there are several windows through which the human tissue can propagate light. And so we can create nanoparticles, in this case metal nanoparticles, that absorb light exactly within this transparency window. And now we are all set to go for the applications. So I was mentioning that this excitation of electric field is essential for the applications that we do, and this is connected to this ERS that you see here. So here you see that the electric field can be excited and confined differently depending on the morphology of the particles. I showed you this already in the previous slide. You can do, and now you can look at what happens in Raman scattering. If you're not familiar with this, it's quite simple. So you excite a molecule with a certain energy of light, and then you excite electrons from the ground state into either an excited state, and this would be electronic spectroscopy, or into a forbidden state, and then there is emission of another photon with a slightly different energy than the incident photon, and this is what we call Raman spectroscopy. Now, this is a process that allows you to monitor the vibrations in molecules, so you can obtain a fingerprint of each molecule, but it is a very inefficient process because of these forbidden states that are involved. And so if you want to use this as an analytical tool, this is not very useful because you need a lot of material to actually obtain information. Now, exactly 50 years ago, it was discovered coincidentally that if the same molecules are located on top of a nanostructured metal surface, you can enhance the intensity of this Raman spectroscopy signal by many orders of magnitude. So it's actually the intensity of the local electric field divided by the initial intensity to the 4th power. So if we are enhancing the electric field here by 100 times, we obtain 10 to the 8th increase in the Raman signal. So we are able to detect even one single molecule with precision and selectivity. And this is what we try to do. And so we can do it either by modulating the morphology of the particles, or we can do it by bringing particles close to each other, because then we create these hotspots at the gaps between the nanoparticles. And so we decided already more than 10 years ago that we wanted to go for real applications. So we proposed to the European Research Council that we are going to target bacterial infections, and not exactly to cure infections, but to understand how they work. So you know that bacterial infections are becoming a competitor to cancer in terms of how badly they affect humanity. And there is also a lot of concern because so many antibiotics are released to the environment that bacteria mutate and become resistant. And so it is crucial to understand how bacteria communicate to each other and decide to create an infection. And this is what is called quorum sensing. So basically, you have a collection of bacteria in a medium, and they start releasing certain molecules, some metabolites, that can be recognized by proteins that are expressed on the surface of other bacteria. So they can actually read 2 minutes, okay, 3, okay, thank you. So they can actually read how many individuals are in the population, and then they decide how to create a biofilm. And so what we did was actually calculating and creating the substrates that allowed us to grow bacteria on top of them, and then by measuring the surface-enhanced Raman spectrum of one of these molecules, we can actually monitor how bacteria, at which moment they decide to create a biofilm. And we also studied, for example, how they talk to each other, how certain types of bacteria can inhibit the growth of others. So this was something that we are still continuing, but we are in a position to actually track how the bacteria are working in a colony. On a second project, we decided to go and try to understand cancer by essentially doing something similar, but in this case, creating tumors in the laboratory. So we are actually creating these scaffolds in which we embed the metal nanoparticles so that we can detect metabolites released by cancer cells, so we can study in real time cancer metabolism, or we can monitor cell death. So in this example that you see here, the green spots are cells that are expressing the green fluorescent protein. They are embedded in these gaps between these lines that are made of a hydrogel containing gold nanoparticles. And then we can irradiate, we can monitor the diffusion of the drug through this construct, and we can correlate the diffusion of the drug with the death of the cells that are embedded in this system. Going further very quickly, we also were able, in this project, to design environments that are very similar to what we have in the human body. They are created by decellularizing real tissue, and then we can grow actual cancer cells surrounded by endothelial cells and macrophages so that we can simulate a real tumor. And then we can go and observe how this tumor behaves over time. For example, watching the onset of metastasis depending on the type of the tumor cells that we have in this in vitro replica of a real tumor. So I don't have time to go into much more detail. We are very excited about this work, and we believe that we are going to, I have to go through this one very quickly. We believe that we are trying to help to go from standard medicine to personalized medicine so that we can monitor the state of a patient and then recommend which will be the most appropriate drug, the most appropriate doses that will cure that specific patient, and maybe include the SCRS test within the standard tests in the hospitals. So I just wanted to thank my lab, funding agencies, and you. Thank you so much.